أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم. Dear student, Assalamu alaikum. I am Dr. Muslim Khan, Assistant Professor, Department of Chemistry, KUST. Today, the topic of our discussion is the first part of electrophoresis. Today is the seventh lecture of a course. There are different questions coming in our mind. That first of all, that what is electrophoresis, and we will discuss also a brief history of electrophoresis. That what have been done so far in electrophoresis technique, who discovered what, and also different types of electrophoresis and electrophoresis techniques, and different principles of these different types of electrophoresis techniques, and also what are the equipment needed and what are the reagent needed in electrophoresis technique. So we will discuss the detail of those equipments and reagents in electrophoresis technique. Agarose gel is used in electrophoresis technique, so we will discuss how we can we pour an agarose gel in electrophoresis technique. So once the electrophoresis is done, we get the bands of those electrophoresis. So what is the meaning of the bands after electrophoresis technique? So what is electrophoresis? Basically, electrophoresis is analytical method, analytical technique, whereby charged molecule in a solution, this is important, charged molecule in a solution, mainly proteins and nucleic acid, migrate in response to an electrical field. Basically, electrical field is applied to the molecules which are in solution. So these charged molecules are moving in response to this electrical field. So the moving of these molecules, what are the factors that the moving of these, the motion of these molecules, these charged molecules depends. The rate of migration of this to the electrical field of these molecules depends on the strength of the field, how strong the field is, how weak the field is. The motion of these molecule depends, and also on the net charge of the molecule, and size of the molecule is very important, and shape of the molecule also very important, and also on the ionic strength, viscosity, and temperature of the medium in which molecules are moving. Temperature of the medium is very important. So we say that an electrophoresis basically due to the due to the due to the application of electrical field, the charged molecules are moving in response to the application of electrical field. But the motion of these molecules depend on certain factors. Thus, that is strength of electrical field you applied, the net charge, size, shape, ionic strength, viscosity, and temperature of the medium in which molecules are moving. So these are the factors in which the motion of these molecules depends. So it means that basically electrophoresis is an analytical technique which is very simple, rapid, and highly sensitive, used for the separation of these molecules, used as a separation technique for the separation of these charged molecule particles. Uh, mm, so it means that this technique is used analytically to study the property of a single charged species or a mixture of molecules. So it can, just like chromatography, like in some of the technique which you want to use to study qualitative or quantitatively or qualitatively some of the molecule, beforehand you are doing basically preparative chromatography. So similarly, you can do preparative electrophoresis is for the for the for the separation of molecule as well. So it can also be used preparatively as a separating technique. Uh, Electrophoresis is usually done with gels formed in tubes, slabs, or on a net on a flat bed. So a gel is very important in electrophoresis. In future slides, I will tell you that what is the purpose of gel used. But I will show you the texture of this tube, slab, or flatbed electrophoresis technique in our coming slides. So in many electrophoresis units, the gel is basically mounted between two buffer chamber. There are basically two buffer chambers. In one buffer chamber, anode is fixed, and another buffer chamber, cathode is fixed. So the gel is mounted between two buffer chamber containing separate electrodes. Means these two electrodes are completely separate from each other in two buffer chambers. The purpose of the gel is the only electrical connection between the two chamber is through this gel, so that they are not mixing with each other. The purpose of the gel is that to electrically connect these two chambers with each other. Uh, so, coming to the history of electrophoresis, brief history. In 1834, you know the famous Michael Faraday laws. Mike Faraday's first law of electrolysis, Faraday's second law of electrolysis. Basically, these two laws were the basis for electrophoresis, for the development of electrophoresis technique. In 1882, Robert Koch developed an agarose culture medium, which is used in electrophoresis. In 1937, Tiselius discovered, made a new apparatus for electrophoretic analysis of colloidal mixture. This was one of the breakthrough in electrophoresis history. In 1946, agarose gel was used for the electrophoresis technique, and in 1955, Oliver Smith used star gels. These are the different gels like agarose gels, star gels, which are used in the electrophoresis technique. In 1957, Joachim Kohn used cellulose acetate electrophoresis technique, and 1950, just after this cellulose acetate electrophoresis, 2 years after, 1959, acrylamide gels was used as a gel in electrophoresis technique. In 1969, Weber, K, and Osborne used SDS gel electrophoresis. We will discuss in future what is that. And also 1971, Dana and Nathan, for the first time, separate DNA by gel electrophoresis, which was one of the major breakthrough in electrophoresis technique. 1977, sequencing gels were used in electrophoresis technique. And also the last one, 1983, which is quite recent, pulse field electrophoresis. We will also discuss maybe in this lecture or the next lecture what is pulse field electrophoresis technique. So also capillary electrophoresis technique, 1983 was also developed. Coming to electrophoresis, so the principle is, so an electrical field, charged molecule, not neutral, charged molecule and particle migrate to the opposite charges, usually in aqueous solution, and usually that is buffer solution is used as a medium for the migration of these molecule and particle towards opposite charges. Due to their varying charges and masses, this is important. Due to the varying charges and masses of these molecules, different molecule and particle in the mixture migrate at different speeds. Means bigger masses, molecule having bigger masses will definitely move slowly in comparison to those molecules which are having smaller masses that will move fastly. So the difference of the movement of because of these massive and varying charges, these molecules separate from each other. So what does it mean? I want to say this thing, that electrophoresis is basically a separation technique. So as a result, separated into a single fraction that is called bands. So this is These are let's say these are the negatively charged molecule having different masses. This is cathode and this is anode. You know that cathode is negatively charged, anode is positively charged. So you put a sample here. So you also applied an electric field. So you pour this buffer solution in and it. So this is a medium. So these are negatively charged. Definitely, these molecules will go towards positively charged anode. So what is this is a bigger molecule having greater mass. This is smaller than this, and this is smaller than this. So this smaller will move fast. So this will reach first here to the anode, and this will like after this, and this will after this. So it means that the difference because of the application of this electrical field, there these ions are moving towards their respective electrodes. These are separated from each other. So this is the main principle of electrophoretic technique. So this migration depends on, as I told you, strength of electrical field. This is just the revision of the last slide. The migration depends on the strength of the electric field, temperature of the medium, uh, also feature of the molecule like size, shape of the molecule. Net charge of the molecule is also very important. Size of the molecule, these are the feature of the molecule. Net charge, size of the molecule, shape of the molecule, feature of the gel also depends feature of the gel, because gel act is electrical connection between these two buffer chamber. Gel type, which type of gel you are using. Also gel concentration, what concentration of gel you are using. uh, and this migration depends also. Buffer type, which type of buffer you are using, what is the pH of the buffer. So depend, like means if you are using acidic buffer, so depends whether these molecules will be negatively charged, positively charged again. So electrophore, it means that electrophoresis separate nucleic acid, proteins, peptides, amino acid, organic acid and bases, drugs, pesticides, inorganic anion cation. These are the measures like mainly separate nucleic acid, proteins, peptides, amino acid, organic acids and bases, drugs, pesticide, inorganic anion and cation. So we can say that everything that can carry a charge can be separated by electrophoresis technique. So, what are the electrophoresis types basically? First of all, which is called gel electrophoresis technique. Depends which type of gel you are using. Agarose gel. So, agarose gel electrophoresis. Polyacrylamide gel, polyacrylamide gel electrophoresis, and some other electrophoresis technique as well. So next one is pulse field gel electrophoresis technique. This is called PFGE technique. We will discuss also this one. And other one is capillary electrophoresis technique. This is also very important. And this term isoelectric electric force focusing electrophoresis is also used. We will discuss this one as well. And also all of these above techniques are 1D electrophoresis, one dimension. And nowadays 2D electrophoresis technique is also used for the separation of the molecule. First of all, what is gel electrophoresis technique? Uh, Use of gelatinous material. Gel is used and gel electrophoresis technique. So the gel act as a support medium in gel electrophoresis technique, used to separate mainly proteins and nucleic acid in this gel electro. So what are the different types of gels that are used in? Starch is used as a gel in a gel electrophoresis technique. Uh, Polyacrylamide proteins, small nucleic acid fragments are used as a gel in gel electrophoresis. Also, agarose nucleic acid, large protein. These two are the main, like these are the commonly used gel types in gel electrophoresis technique. And the last one is cellulose acetate protein gel is also used. Cellulose acetate, which is called cellulose acetate electrophoresis technique. So the first one is agarose gel electrophoresis. This is agarose specificity. This is the structure of agarose. You can see it here. So this is technique is easy, very fast, and well-established method for the separating DNA fragments. This agarose gel electrophoresis technique. Agarose, this is a polysaccharide derived from the seaweed, this agarose, and this beta 1,3 D galactose, uh, uh, 3,6, and hydro alpha 1,4 galactose. And this dissolve in a boiling water and hardens, become gel when cooling. So basically this molecule is put in a boiling water, it becomes hard, and it becomes gel when you are cooling this. So the advant this is having a bigger pore size than polyacrylamide. Um, mm, it can be advantages sometimes, but can be disadvantages sometimes as well. So whenever you want to separate, as I told you in last lecture, this one, that this is well-established method for DNA fragment. So whenever you want to separate DNA fragment, this agarose gel concentration, this is just this was just for whenever you want to do DNA separation, you must know that how much concentration of agarose I should use. This is just like point 0.3 for this size of DNA fragments, and zero point 0.6 for this size of DNA fragments, and zero point 0.7 for this size, and zero point 0.9 for this size. So depends that which size of DNA fragments you are basically separating. You should use that concentration of agarose gel in your experiment. So the next one is a page, polyacrylamide gel electrophoresis. This is acrylamide. This is acrylamide, and this is treated with this molecule, and this become polyacrylamide, means become polymer. This is a synthetic polymer, as I told you. This is a polymer, and formed from acrylamide subunit. This is acrylamide subunit. This formed from this, and acrylamide with a cross linker, methylene bis acrylamide. This is cross linker, and polymerization catalyst is used, and this ammonium persulfate plus tetramethyl ethylene diamine and light, and 3.5 to 20% concentration, and high resolution. And acrylamide is one thing you know, that acrylamide is a dangerous neurotoxin, so you should be careful of using this acrylamide while doing your experiment. So buffer is also used in electrophoresis. What is the purpose of a buffer? So it provides ion in a solution for electrical conductivity. This is used as a medium. This aqueous buffer is used as a medium and for electrical conductivity. So it prevents the pH changes, because once the pH is changing, then at a change pH, the molecule, the charged particles are basically, the charge is changing then, because at a pH 7, at a pH 9 or pH 3, so they will be in a different form, ionic form. That's why it prevents the pH changing. Also, it needs, it provides the electrical conductivity as well. So common using buffers are tris borate ADTA stable, expensive. This but long separation time. This is used for page. Tris acetate EDTA, inexpensive, short separation time is selling. And tris phosphate EDTA in RNA. Sodium phosphate buffer is also used, and these are the other buffer solutions which are used in electrophoresis technique. So what are the equipment needed in electrophoresis technique? Power supply is one of the main. Cooling apparatus, and electrophoresis gel apparatus, vertical or horizontal, depend. White light, UV light box, digital camera, gel documentation system, and reagent, gel staining chemical, preparation gels, gel chemical, buffer, loading dice, and other laboratory like pH meter, pipette, laboratory scale, and stair plates. These are the equipment used in the electrophoresis technique. So in our next lecture, we will discuss some more aspect of electrophoresis technique.